This location? We are centrally located in Europe and we have the world's best infrastructure. Oh, and the train is always on time, so let's take a journey. Did you know that Switzerland has 4 official languages? Ja? No? Forse? Welche dia? Don't worry, we also have excellent English skills. However, we don't spend all day chatting. We love to work. In 2012, we could have voted for longer holidays, and we voted against them. It's crazy, isn't it? But we're not crazy. Our economy and government are among the world's most stable. That, combined with our outstanding talent pool and a deregulated labor market, makes Switzerland the most innovative and competitive nation in the world. Our Swiss dual education system provides an excellent mix of both vocational education and apprenticeships. Now let's take a look at industry. Maybe Germany, the United States, and China are well known for their industrial output. But did you know that Swiss industrial production per capita beats all of them? Sorry, guys. Our country is small, but hey, 3 Swiss companies are among the global top 20 most valuable companies. Furthermore, we offer the best quality of life. That's why 3 Swiss cities are among the world's best for the highest quality of living. In addition, Switzerland is one of the happiest nations on the globe. Perhaps because we eat on average 12 kilograms of chocolate per year? Switzerland is the best location for innovation, has stable political, economic, and financial conditions, has the highest living standard, and welcomes your business. So tell us now, when will you invest in Switzerland? Switzerland Global Enterprise, enabling new business. Find out more at s-ge.com.
Karnataka has the right ingredients of all that it takes to build a very advanced biotech ecosystem.
Any road that leads out of Bangalore, or for that matter into Bangalore, is actually, there is a biotechnology hub.
The ecosystem in Karnataka, especially in Bangalore, is very, very industry friendly. For biotechnology, we have high-class research institutes, whether it is the Indian Institute of Science, or whether it is the NCBS, or is the Agriculture Sciences University, or the IBAB.
The Bangalore Life Sciences Cluster is present at the University of Agricultural Sciences campus, and it's primarily made up of three institutions: the National Center for Biological Sciences, the Institute for Stem Cell Biology and Regenerative Medicine, and the Center for Cellular and Molecular Platforms called CCAM. Together, they become an outstanding intellectual hub.
The uniqueness of the Bangalore Helix is the presence of academic institutions like IBAB, Center for Human Genetics, as well as the Bangalore Innovation Center.
IBAB is a first of its kind institute involved in training and combined with biotech students in cutting edge areas of bioinformatics, and it is involved in incubating young companies and helping entrepreneurs in these areas.
The breadth and the depth of the skill base that is available here. I don't think there is any other city in India which has systematically, over the past 30 years, built up such a broad base of people who, in all areas of biotechnology.
So whether it is molecular and cellular biology, whether it is genetics and genomics, whether it is high-end IT and big data analytics, or whether it is high-end engineering and bioprocessing and biomanufacturing, Karnataka has it all.
The Karnataka Biofuel program is very unique. It empowers the rural community. It strengthens the agricultural activity in the rural areas. It generates a whole lot of employment through value addition activities in the rural areas.
One of the benefits of our strand virtual liver based approach to studying toxicity is that it actually minimizes or even eliminates in some cases the use of animal models, and therefore in where you do not want to have animal testing and yet want to understand the toxicity of molecules. This is perhaps one of the few options available.
The Center for Human Genetics is trying to fill this niche area, which is a highly demanding area, because under one institutional framework you need to have both medical specialists as well as basic scientists working together and complementing each other's skills.
We have got lots of scientists and doctors who are able to provide high-class care for these patients who have got extremely rare disorders. This has been possible because of our partnership with the biotech industry, which has helped us in providing enzyme replacement therapy to the patients and also the special foods that some of these rare disorders require.
The cancer care has undergone a radical change in the last 5 years. From evidence-based medicine, we have come to personalized medicine. And this has happened mainly because of better understanding of the disease, better understanding of the gene makeup, and better therapy on an individual basis suited to that based on their genetic expression and the biomarker expression. So this will revolutionize cancer care, and we in India, particularly in Bangalore being the hub, can truly become leaders, global leaders in the genomic therapy of oncology.
Karnataka has supported us in this journey for the past 30 years by providing us the conducive climate to do business here in India, and we have succeeded.
The kind of work that we do involves a lot of collaboration. And in Bangalore, we have found really the kind of collaborations, whether it's with big companies or smaller companies, who which really are very relevant for our kind of work.
The strengths which IBAB gave us was the laboratory space, necessary infrastructure needed for a startup biotechnology company, and actually mentorship, which is more needed in this field.
IBAB offers also several other benefits like a wealth of very talented young people that we can choose from, interns, and also great infrastructure that the institute provides.
String is an early stage startup. We are developing an innovative solution to take waste, organic waste particularly, to biodegradable plastics. We've really built the team and the product at a remarkable speed over the last year, and the intellectual environment that Bangalore provides for a startup such as ours.
There are more and more entrepreneurs coming, and as the ecosystem grows with all the components already in place, any entrepreneur today finds it very easy to come and set up his operations here, and there is mentorship available, other business-friendly policies, the ambience is there, and so I see a very bright future for Karnataka's biotechnology industry.
The government of Karnataka decided to set up an autonomous vision group for biotechnology, which was an optimal blend of academia, industry and the government. The harmony and the alignment that these three groups of people have had at this vision group has really enabled us to come out with some of the most pragmatic policies in biotechnology, the required investments in biotechnology from the government, which has really allowed us to take a unstinted leadership position.
The state has come out with always business-friendly policies, and in and around Bangalore there are a number of higher education institutions which provide the talent pool for such industries or enterprises.
Investing in quality foundations in science and technology is vital so that you're prepared for whatever change the world brings about, and so you can grab that change and do well.
There's a very, very unique ecosystem in this part of the world, and great things are expected to happen, and all this has been made possible solely because of the proactive role that Karnataka government has played.
It really doesn't matter which area of biotechnology you're working on. I think this is the best place to be.
Ideas need entrepreneurs. Entrepreneurs need startups. And startups need a robust ecosystem to emerge as an unicorn. This ecosystem is what makes Karnataka the innovation and technology hub of India. Government of Karnataka supports startups right from offering physical space to setting up of incubation in partnership with NASSCOM, IAMAI bodies with state-of-the-art facilities, amenities and support services. Mentorship by industry leaders and domain experts to advanced tools for testing, instrumentation, industry connects, investor connects and global connects. Filing patents and protecting ideas to providing mentoring support and brand development assistance. The government of Karnataka has come in to assist budding companies through every hurdle and provide all kinds of resources across all its physical infrastructure. With dedicated facilities and state-of-the-art infrastructure all under one roof, exclusively for IT and BT startups. But only physical space is not enough to house 8,000 startups. Karnataka is also the first state in India to come up with sector-specific policies and adapt a framework for innovation engagements at a global level with countries from around the world to assist solve a collective need for nations world over. A space where this all comes together is the Bengaluru Tech Summit, a conference that brings together innovators, startups, investors, government, policy makers, academia to foster collaborations, co-creation, co-innovation and co-invention. Some of the largest companies in the world have set up their own incubation centers in the city of Bengaluru. Not only is Bengaluru one of the easiest places to find skilled team members for a startup, it is deemed as the startup capital of India with more new startups incubated here than anywhere else in the country. To give a thrust to the emerging sectors and sunshine technologies, the state government, through a joint initiative with corporates and MIT, has established multiple centers of excellence to boost the accelerator support to the entire startup ecosystem. In addition to the COEs, the government of Karnataka is supporting establishment of 5 technology business incubators in institutions of higher learning with well-developed R&D facilities to foster a strong link between R&D and commercialization of technologies developed. And the city is home to 14 of India's 33 unicorns and 20 soonicorns, which is highest in the country. Bengaluru leads in the startup race when it comes to the number of funding deals, with Bengaluru-based companies having raised $2,436 million in the first half of 2020. The drive to grow and innovate is not restricted to Bengaluru alone. It goes beyond Bengaluru to meet this growing entrepreneurial spirit in the state and to nurture innovation at district level. The government of Karnataka has set up New Age Innovation Networks, NAIN, in the emerging clusters. NAIN envisages and encourages student entrepreneurship at a grassroots level, while also setting up R&D labs and common instrumentation facilities in cities across the state, including Mysuru, Hubballi, Dharwad, Belagavi and Bagalkote. Additionally, the state has set up 5 common prototyping or instrumentation facilities in Jalhalli, Mysuru, Mangaluru, Shivamogga and Belagavi, fueling the startup ecosystem through innovation hubs in Tier 2 cities, thus empowering hardware startups across the state. The government of Karnataka is the only state supporting early stage startups by Idea to POC or Elevate Grant in Aid to identify innovative startups across the state and elevate them to the next level. Elevate Unnati is an Idea to POC Grant in Aid for startups promoted exclusively by SC/ST entrepreneurs of the state. For social innovation, Karnataka uses a model of grand challenges to focus attention and effort on specific problems. The government has tied up with Catalyst for Women Entrepreneurship for promoting entrepreneurship among women. Kitwin Fund focused on encouraging entrepreneurship in the state, promising startups in need more than capital or financial backing to succeed. The government also incentivizes startups through reimbursement of GST, marketing incentives, patent filing costs, and provides booster kit in form of web credits. Between the schemes, policies, partnerships and alliances, Karnataka is geared to grow faster than ever. The state is also ready to establish an AI and robotics technology park and creating a digital economy mission to leverage the 500 plus R&D centers to retain Karnataka's position as a leader in innovation-led economic growth. The state also launches a first-of-its-kind sector-agnostic regulatory sandbox for promoting upcoming technologies. The government of Karnataka is also planning a new and renewed startup policy in 2021 that will establish Karnataka as a power player in India's growth as a global innovation leader. After all, startups without an ecosystem are just entrepreneurs with ideas. And our startups are fully backed by the government ecosystem that fuel the innovative spirits of the entrepreneurs and risk appetite angel investors and VCs.
Odisha is taking many initiatives for industrial growth in the state and has turned out to be a preferred investment destination in the country. The major investors in the state are NTPC, MCL, SAIL, Tata Steel, TCS, Adani, JSPL, PPL, IOCL and many more. More than 300 companies have invested and the state has received investment worth more than 5 lakh crore. Major investment sectors are IT, fertilizer, petrochemicals, food processing, health, infrastructure, ESDM, metal and minerals, manufacturing, textile and tourism. We are aware that Indian biotech industry is likely to experience significant growth on the back of increasing economic prosperity, health consciousness and billion plus population base. Odisha is now single-mindedly focused on building a successful startup ecosystem to facilitate the biotech industry. The State Industrial Policy Resolution 2015, IPR 2015, recognizes biotech sector as a priority sector and extends all the fiscal incentives, special incentives and other support measures to the biotechnology sector. In such cases, the industrial unit can choose to avail a particular incentive under this policy or the sectoral policy like agriculture policy, food processing policy, MSME policy, startup policy, SEJ policy, etc. Odisha has also come out with a biotech policy in 2018 which assures incentives at par with IPR 2015 and also supports biotech R&D ecosystems through supporting biotech incubation centers of excellence and also by supporting early stage funding like immersion fellowship and bio-innovation grants. The state has set up Industry Infrastructure Development Fund with an initial corpus of 100 crore. The IIFDF will be utilized for development of quality infrastructure such as roads, power, water, waste management, etc. Government of Odisha has partnered with Bharat Biotech to establish a biotechnology park at Bhubaneswar. An incubation center has also come up at ILS Bhubaneswar with the support of Government of Odisha as well as Department of Biotechnology, Government of India. Odisha possesses a unique model of connecting academics, industries, R&D institution and startups. At the same time, the unique bioresource in the field of agriculture and biomaterials in Odisha, particularly in the tribal areas as well as large marine and coastal region of the state, also will enable value addition and product development through biotechnology applications. Odisha, with its bio resources, and Bhubaneswar as state capital with large number of scientific institutions will enable academician collaborations with potential industries and will engage the trained human resources for the purpose. I hope with cooperation from potential investors, Odisha will definitely develop into a preferred investment destination in the biotech sector and contribute to better lives and livelihoods. Thanks you all.
In healthcare, a vast ecosystem is helping therapy innovators become more patient-centric and more effective than ever before. Cytiva supports researchers, biopharma, and clinicians in the pursuit of more targeted treatments. In discovery, speed and accuracy are everything. The faster and smarter researchers can work, the more patients benefit. To ensure that new, more targeted therapies become reality, we're equipping biomanufacturers with the tools and expertise they need to produce cost-effectively and quickly. How can tens of thousands of individualized patient doses be manufactured quickly, efficiently, and securely? The answer, with a healthy dose of expertise and innovation. We're creating processes and solutions that are scalable and address complex needs. We work to deliver diagnostic tools and other technologies to bring the right therapy to the right patient at the right time, so that life-saving medicines come within the reach of more people and could save more lives. Cell culture.
I wish I had some music in the background or something.
That's totally fine.
This is eerie quiet.
Yeah, this is weird that there are no in-person meetings.
We are live. We are live now.
Okay. Hello, everyone. Good morning and good evening to the people who are on the other side of the continent. My name is Sita Avasti. I'm a research associate professor in the Infectious Disease Division at University of Pennsylvania. I welcome you all at the Global Bio India 2021. It is my honor to moderate this session on mRNA vaccines. How timely it is. While the concept of using mRNA to synthesize protein is logical, but it required decades of painstaking research to overcome the series of hurdles. For example, like figuring out how to modify RNA so it is not quickly degraded by our own cells, or how to produce it so the immune system will not react adversely to us, and induce really large amount of desired protein. And finally, how to deliver mRNA to the human body. So when pandemic hit in 2019, about 30 years of meticulous basic research allowed companies like BioNTech, Pfizer, Moderna to brought mRNA vaccine technology in record time to prevent people from falling seriously ill or dying. So in this session, we will hear stories from experts, from academics, industries, and regulatory agencies on aspects of mRNA vaccine technology and beyond. And with that, I'm going to introduce our first speaker of this session, Dr. Norbert Purdy. And it is my absolute pleasure to introduce him to kickstart this session. Dr. Purdy received his doctoral degree in biochemistry and genetics. He then joined University of Pennsylvania in 2011, where he worked with Dr. Drew Weissman and collaborated with Dr. Kathleen Carrico. With Dr. Weissman, he made seminal discoveries in nucleoside-modified mRNA technology with a particular focus on developing new generation of vaccines for emerging diseases. He went on to find immune mechanisms to explain why modified mRNA is such a robust platform for vaccines and immunotherapeutics, and published numerous milestone papers on the topic. So without a doubt, I can say Dr. Purdy is a true pioneer of the modified nucleoside mRNA vaccine technology. So my friends, I present to you my friend, colleague, and collaborator, Dr. Norbert Purdy. The floor is yours, Norbert.
Thank you very much, Sita, for the very kind introduction and the invitation. It's really my great pleasure to give a talk today. Good evening, everyone. So I will be telling you about very briefly the history of nucleoside modified messenger RNA, therapeutics messenger RNA vaccines, and then I'm going to highlight some of the applications. Of course, it won't be possible to talk about everything, but I will just mention two examples today. So the very first paper that demonstrated or suggested that in vitro transcribed RNA could potentially be used for therapy was published in 1990. This was a beautiful science paper in which the authors injected mice intramuscularly with in vitro transcribed messenger RNA encoding reporter genes, and protein production from the RNA was clearly demonstrated. I think this was a very important discovery, but despite of the important findings of this paper, most people remain skeptical about the therapeutic potential of RNA, and the field focused on other approaches, for example, DNA-based approaches. The main concerns were the unstable nature of messenger RNA, its really high innate inflammatory capacity, and very low in vivo translatability. But due to the technological advancements and very active research in the past 10 to or 15 to 20 years, these issues were resolved, and messenger RNA, in vitro transcribed messenger RNA, became a very promising new therapeutic modality. Excuse me. I'm going to tell you about some of the most critical discoveries in the past 15 years. So as Sita mentioned, Katalin Karikó and Drew Weissman are really the pioneers of this technology, and they published a groundbreaking paper in 2005. I think this is the most interesting or most critical experiment that I'm showing you in this slide. So what they did, Katalin and Drew isolated RNA from different sources, from mammalian cells, from prokaryotic cells, and then they transfected monocyte-derived dendritic cells with these different types of RNAs, because these cells are very sensitive and they are equipped with many different types of RNA sensors. And most importantly, they included a conventional, unmodified in vitro transcribed RNA in this experiment. And the take-home message is basically the title of this slide. Natural RNAs are not equally potent activators of the immune system. As you can appreciate, the bacterial RNA induced a lot of TNF-alpha production. Mitochondrial mammalian RNA also induced TNF-alpha production. But for example, transfer RNA did not induce any inflammatory response. And most importantly, as shown with the purple bar, in vitro transcribed RNA induced lots of TNF-alpha production. And the big question was why this was happening. What is the difference between different types of RNAs? But Katalin and Drew figured out that the most probably the critical difference is the natural modifications of RNA. We all know that RNA has 4 basic building blocks, but actually you can modify these building blocks. And the most abundant modification is pseudouridylation. Pseudouridine is made by pseudouridine synthase from uridine, and it's very abundant. You can find it in ribosomal RNA, transfer RNA, both in prokaryotic and eukaryotic cells. And indeed, when Katalin plotted the percentage of nucleoside modification in RNA against the immune activation after transfection with these RNAs, it really turned out that the higher percentage nucleoside modified, the higher the percentage of modified nucleosides in the RNA, the lower the immune activation. So if you remember, transfer RNA did not induce immune activation, and actually it contains almost 25% modified nucleosides. But when they transfected with an unmodified in vitro transcribed RNA, it induced lots of inflammation. So when they got back and they repeated these studies, the transfection of monocyte-derived dendritic cells with unmodified and different types of modified RNAs, it really turned out that unmodified RNA induced lots of TNF-alpha and IL-12 production, and the nucleoside modified RNAs did not induce such a high amount of pro-inflammatory cytokines. So the take-home message was that modified nucleosides can decrease immune activation, which is extremely important for the safety of messenger RNA-based therapies or vaccines. A number of years later, they had another important discovery. It turned out that incorporation of modified nucleosides in the RNA not only decreases immune activation, but it also increases the translatability, so protein production from the RNA, which is also very important for therapeutic applications. So in this study, they transfected cell lines or primary cells, murine dendritic cells, with renilla luciferase encoding mRNAs, either unmodified or modified, and then they just measured luminescence, which reflects active translation from the RNA, and it really turned out that, for example, pseudouridine-containing RNA there was a much higher amount of protein production from the pseudouridine-containing RNA. So this was another very important discovery. When I joined Katalin Karikó and Drew Weissman in 2011, we could really make high-quality RNA. This was not the problem. The problem was that we did not really have a safe and efficient delivery material for in vivo RNA delivery. So we could use mRNA transfection reagents for cell transfection studies, and they worked very well, but all of these reagents are very toxic or just inefficient when you want to use them for in vivo studies. So we spent years on trying to find these lipid nanoparticles for in vivo delivery, and after 3 years, we found a company called Acuitas Therapeutics, and we asked them to formulate our messenger RNAs into their special lipid nanoparticles. These lipid nanoparticles have 4 components. They self-assemble to 60 to 100 nanometer sized particles, and they were originally developed for siRNA delivery. And so we really wanted to try it with messenger RNA, and asked them to formulate some firefly luciferase encoding RNA into these LNPs, and they did that, and we tested them after injecting mice intravenously, intraperitoneally, intramuscularly, subcutaneously, intradermally, intratracheally, and just measure protein production from these mRNA LNPs by in vivo imaging. And it really turned out that these lipid nanoparticles worked very, very well. We saw 2 different types of kinetics. When we injected mice intradermally or intramuscularly, most of the protein was produced at the injection site, and for actually a pretty long duration, for 8 to 10 days. So this is a very good kinetics very favorable kinetics for immunization studies. When we systemically injected the mRNA LNPs, they targeted primarily the liver, and the liver cells produce tons of protein, but only for a shorter amount of time. So we call this platform nucleoside modified mRNA LNP platform, and this is being used by Moderna and also Pfizer-BioNTech. So in both SARS-CoV-2 mRNA LNP vaccines, very similar platform is used. So how do we make the RNA? The RNA is made from a plasmid DNA template, and we perform an in vitro transcription reaction. After the in vitro transcription, the RNA undergoes a very stringent purification procedure to get rid of the double-stranded RNA and other contaminants that would otherwise recognize the pathogen-associated molecular patterns by the immune system and would induce unwanted immune responses. And like I told you before, we replace uridines with 1-methyl-pseudouridine that make the RNA more stable, highly translatable, and less inflammatory. And once we make the RNA, it gets formulated into lipid nanoparticles. And so this is the name of the platform. And like I told you, we use this platform for many, many different purposes. And I just listed some of the most critical advantages. The development of these vaccines are very fast, as demonstrated by Moderna beautifully. They developed a SARS-CoV-2 vaccine in 42 days after obtaining the the sequence of the spike glycoprotein of SARS-CoV-2, and these vaccines work incredibly well in people, and now tens of millions of people received the Moderna or the Pfizer-BioNTech vaccine. So I think proof of concept was very clearly provided. The very first paper that demonstrated that the nucleoside-modified mRNA LNP platform is a really good vaccine was published actually almost exactly 4 years ago. So we generated a vaccine against Zika virus. The vaccine encoded the pre-membrane and envelope glycoproteins of Zika, and we immunized mice and monkeys with a single immunization of Zika virus vaccine, and the vaccine worked incredibly well. Animals generated neutralizing antibodies very quickly, and these vaccines proved to be protective. There was sterilizing protection in mice and almost sterilizing protection in monkeys. And after we published this paper, several other papers, and now like dozens of papers confirmed our findings, not only for Zika, but also for other infectious pathogens. I would like to tell you very briefly about another potential application. This is not active immunization, but passive immunization using the nucleoside-modified mRNA LNP platform. So as you all know, monoclonal antibodies are one of the fastest growing classes of pharmaceutical products, but their potential is limited by the high cost of development and manufacturing, and we believe that we can use mRNA encoded monoclonal antibodies for therapy, and this would probably be a much more inexpensive alternative for immunotherapy. So we did a proof of concept study in which we used an messenger RNA encoded broadly neutralizing anti-HIV antibody called VRC01. So we generated two messenger RNAs encoding the light chain and the heavy chain of VRC01 and co-formulated these into lipid nanoparticles, and then looked at the kinetics of antibody production in mice. And we also wanted to see if these antibodies are functional and they could protect mice from HIV infection. Animals received a single intravenous injection with VRC01 mRNA LNPs, and we could measure very high levels of antibody as early as 24 hours post-injection. And so basically the construct worked very, very well, and to demonstrate that the antibodies can really protect mice from infection, we used NSG humanized mice that have no B cells, but they have circulating human CD4 positive T cells, so they can be infected with HIV. The experimental setup was very simple. Animals received a single injection with the VRC01 mRNA LNPs or negative control firefly luciferase mRNA LNPs, and 24 hours later, they were challenged with a HIV strain called GRCSF, and we looked at viral RNA levels 1 and 2 weeks post-challenge. Like I showed you before, 24 hours post-injection, animals produced very high levels of VRC01 antibodies, and those animals that received the VRC01 mRNA LNP, they were protected from viral challenge, and those animals that received the control firefly luciferase mRNA LNPs, they developed viremia. So basically this approach worked very, very well, and companies including Moderna, BioNTech, CureVac, they are also very much interested in developing mRNA encoded monoclonal antibodies, and this is also already in clinical phase. So I think my time is up, and then I would like to conclude, and I just want to mention to you that many people helped me in these studies over the years, and I'd like to thank for them, and of course I'd like to thank for your attention, and I'm not sure if we have questions, but I'm happy to take if there is any. Thank you very much.
Thank you, Norbert. I'm going to just start asking you a simple question. I know we don't have much time, but in the beginning, when you were working on it, how did you figure out that in any given condition, mRNA will not be integrated into the human genome? although I understand the biology behind it, but in a simple language that many of our listener can understand and have not that fear. If you can answer. Sure.
Messenger RNA, just again, based on, I cannot really give you a longer answer than that. It just cannot integrate into our genome, and like by definition. So this shouldn't be a concern, and I agree with you. It has to be very clearly communicated that this is not a concern with messenger RNA therapies under any conditions.
Mm-hmm. Thank you. I think we can start our second talk, which will be given by Dr. Andrea Corfey. And he received his doctoral degree from the Institute of Structural Biology at Grenoble, France. He then joined Professor Don Wiley's group at Children's Hospital in Boston, Massachusetts, where he made significant discoveries in the structure and the function of human herpes viruses. Dr. Corfey then made a big move to the pharmaceutical companies with a special focus on vaccines like Merck, Novartis vaccines, GSK vaccines. And in more recent year, 2017, he joined Moderna. And he serves as a vice president and head of research for infectious disease. He led the development of a successful Moderna COVID-19 mRNA vaccine that is now approved for emergency use by many countries and now saving lives of many. Here I present to you Dr. Corfey. You're mute.
Happens all the time, doesn't it? Thank you again, Nasita, for the introduction and for the opportunity to present at the BioIndia. I'll share my slides. I hope you can see it. Give me just okay. And so what I will be doing, I'll give it just a short. Okay, I hope you can see the slides now. I'll give a short introduction on the Moderna mRNA technology, and then I'll focus on really the development of the vaccine against the SARS-CoV-2 virus. And as Dr. Pardy explained, the technology is based on the delivery of a messenger RNA to the cytoplasm of cells, where the mRNA can be taken up by ribosome and translated into proteins. And you can generate proteins in the different formats. They can be intracellular, membrane-bound, or secreted. And therefore, the technology offers many opportunities in terms of really disease that you can target. And clearly, if you express a protein that is a foreign protein, like a viral, a bacterial, or a protein from a parasite, this will result in an immune response. And therefore, the technology is amenable to vaccine development. In the context of the Moderna mRNA technology, the two key elements, of course, are the messenger RNA and the LNP. The mRNA is a standard mRNA molecule. It's capped at the 5' end, has a two untranslated region at the 5' and 3', a polyadenylene tail in the C-terminal part. Sorry, I'll try to put the pointer here. And then between the two untranslated regions is the open reading frame. And the sequence of the mRNA is optimized to result in optimal translation, protein expression, and also stability, which is an important, of course, a feature. One modification is what has been described is the substitution of a uridine with the pseudouridine to prevent excessive innate immunity and therefore dampening of protein expression. The other key element, of course, is the lipid nanoparticle that protects the messenger RNA, but is also critical for delivering of the mRNA to the cells and for escape of the mRNA from endosomes. And there are different elements that are critical. I mean, in the Moderna LNP, we have our own proprietary ionizable lipid, but sterol, PEG lipid, and phospholipid are critical both for the stability, for the release of the mRNA once in the cells, and as I mentioned, depending on how you produce the LNP, that might impact, certainly the efficiency with which the mRNA is delivered to cells, but also the cells that can be targeted. So we consider this as a platform technology because you start from a DNA, and then through an in vitro reaction, you can transcribe the DNA to mRNA. You just need enzymes, a polymerase, buffer, and nucleotides, and then you generate the mRNA that encodes for the protein of interest. Then this is mixed with the lipids and cholesterol and the other elements, as I explained, to form a mRNA lipid nanoparticle complex. And this is basically really the vaccine that can be injected through an intramuscular injection. Now, what is critical is that if you're trying to change the vaccine that you want to make, the only thing that you have to modify is the sequence of the messenger RNA, because everything else remains the same, both in terms of reagents and in terms of the processes. So this allows for a fast development and makes the technology well suited for a pandemic response, as in the case of the SARS-CoV-2. Even before working on this vaccine, Moderna had advanced a number of other candidates, both in the infectious disease space, but also other therapeutics. And in cyan, here are all the targets that we have advanced to the clinic so far. I want to mention that we started with work on flu that was more as a proof of principle that you can actually elicit immune responses in humans. But since then, we have started work on other respiratory viruses, such as our RSV and HMPV, on herpes viruses, such as a CMV and EBV. The CMV program is now in phase 2 and going to phase 3, and also more complex antigens, as such, viral-like particles in the context of the Zika vaccine. And the SARS-CoV-2 was our last entry, though we had performed work on other coronaviruses previously. I want to also mention that we, in the context of infectious disease, we are also advanced to the clinic a gene-encoded antibody, as a party was mentioning, and we have demonstrated that you can express protective level of that antibody in humans. So now I'll focus the rest of the presentation on the development of the SARS-CoV-2 vaccine. And in this case, what we are expressing as an antigen is the spike protein, stabilized by the introduction of 2 proline mutations that stabilize the molecule in the so-called profusion conformation. And this has been a fast development, but it's important, and I'll mention some of the key dates, but it's important to remember that during the development, we did not skip any of the clinical steps. So we performed phase 1, phase 2, phase 3, as for any other programs. In this case, there was a large investment from the US government that allowed us to really start manufacturing and preparing for the next steps, even at risk, even without having all the other data, so a financial risk, if you wish. And then, of course, the support from regulatory agencies that really were very responsive and helpful in moving the program forward. And the key dates that I want to mention is really the 63 days to get from the sequence of the virus to the phase 1 study that was conducted by NIH. So within 43 days, we really had already the vaccine vialed, and that had passed sterility, all the analytical testing, including sterility. So this is really the fastest development that I'm aware of. And since then, we of course we got funding from BARDA for the development of the vaccine. And by end of May, we started the phase 2 study. And 2 months later, end of July, a large efficacy study, a phase 3 with 30,000 subjects. And of course, we got efficacy by end of November. And in December, the VRBPAC recommended that the vaccine was to be approved for emergency use authorization. So There are two parallel tracks. There was the clinical, phase 1, phase 2, and phase 3, but at the same time, the non-clinical studies with immunogenicity, protection, and safety data that were critical to advance the program also through the different steps of the clinical program. And here I'll describe the most relevant data for the non-clinical and then the clinical studies. So we performed initial experiments in mice where we showed protection by the vaccine, mRNA 1273, and that pushed us to test the vaccine in non-human primates, mainly because in this model that had been shown to, NHPs can be infected by the SARS-CoV-2, they develop a mild pneumonia, so it's a suitable model for testing SARS-CoV-2 vaccines. And the monkeys have an immune system that is more similar to humans. You can test also doses of the vaccine that are more, closer to what you tested than in a human study. So the animals received 2 doses, 4 weeks apart, and then 4 weeks after the second dose, they were challenged. We tested 2 doses of the vaccine, 10 and 100 microgram. 100 microgram is the dose then that advanced to phase 3. And we showed that the vaccine was immunogenic. We had the dose response with 100 micrograms raising higher titers of antibodies than 10. And the second dose also boosting the antibody response. When we also looked at the functionality of these antibodies, we could show that both 100 and 10 micrograms we had antibodies that compete binding of the RBD domain, which is the domain of the spike protein critical for binding to the cellular receptor. So these antibodies compete with the RBD-AC2 binding. And the titers of these antibodies are higher than what observed in convalescent sera, so sera from subjects that have recovered from a SARS-CoV-2 infection. And a similar pattern was observed when we looked at the neutralization in a live virus neutralization assay, with titers, again, that were superior to what's seen in convalescent subjects, suggesting that the vaccine could confer protection, although not demonstrating it. So to show that really the benefit of the vaccine, we looked at a replication of the virus post-challenge in the lung and in the nose. This is done by looking at the subgenomic RNA that is really a marker for the replicating virus. And you can see that in the lung, at both 10 and 100 microgram, basically day 4, there's no virus that can be, replicating virus that can be detected. At 10 micrograms, we had only 1 animal. And a similar pattern is seen in the nose. At day 2 and 100 micrograms, there is no replicating virus. At the 10 microgram, only 1 animal had the still replicating virus at the day 4. So, of course, the protection seems to be stronger in the lung than in the nose, and this has been seen also for other vaccines. But basically, this demonstrates that, at least in this model, the vaccine is protective in the lung and partially protective in the nose. We also performed experiments in hamsters, and the reason is that hamsters develop disease or more clinical symptoms. In this case, you can monitor for weight loss, something that you don't see in non-human primates. And we showed that at 3 different doses, 1, 5, and 25 micrograms, with 2 doses, that the vaccine elicits neutralizing responses that protects from weight loss at all doses tested, and also that really the vaccination and the immune responses are able to clear the virus in the lung and the nose really quickly, and by day 4 there was no really virus in the detected. So I'll move now to the clinical studies. As I mentioned, we performed all the studies needed, phase 1, phase 2, and phase 3. The phase 1 is to really show safety and immunogenicity. Then phase 2, we confirmed the dose, and phase 3 was a large clinical study for the efficacy. And then we have a number of other studies that are ongoing. For instance, the pediatric study to really go down with age to infants. So in the phase 1, we tested the vaccine in 3 age cohorts, 18, 55, 56, 70, and above 71. 3 doses, 25, 100, 250. By the time we got to the elderly, we realized that 250 was too reactogenic, so we tested only 25 and 100 micrograms. So and the vaccine was administered as 2 doses, 1 month apart. And here I'm just showing the immunogenicity in terms of neutralizing responses across age cohorts, and only for the 100 microgram dose, that is the dose that we then selected for the phase 3 study. And you can see that after 2 doses, all the subjects responded to vaccination, that the immunoresponses, neutralizing responses as measured by in a pseudovirus neutralization assay are consistent and similar across age strata and above what's seen in the median for the convalescent subjects. And this is important because it suggested that the vaccine could potentially protect also the elderly that are severely affected by this virus. We also looked at the T-cell responses, and we saw again that in all the age cohorts, there were strong CD4 T-cell responses, Th1-based responses. So no Th2, which is a concern for respiratory enhancement of respiratory disease that has been observed in some older inactivated viral vaccines. And we wanted to exclude that this could be a problem in the context of the SARS-CoV-2 vaccine. So a Th1 biased response really suggests that the vaccine should be safe and not trigger any ERD. So now I'll mention the phase 3 study. Was 30,000 subjects, was an efficacy study, 50,000 subjects in the placebo, 50,000 in the active vaccinated group. And what we looked at as a primary endpoint was prevention of symptomatic disease. And so we had an interim analysis and then also then the primary analysis with 196 subjects. And what we demonstrated that the vaccine is highly efficacious and prevents symptomatic disease with an efficacy of 94%. This was the primary endpoint, but also we looked at the severe disease. And as a part of the secondary endpoints, also we are looking at prevention of infection and other endpoints. But I would like to highlight that in the context of prevention of severe disease and therefore hospitalization and death, the vaccine was extremely efficacious, and that the primary analysis, there were 30 cases in the placebo group and none in the vaccinated group. So again, suggesting that the vaccine has high efficacy. So in the interest of time, I will move to the conclusions. The vaccine has shown to be highly efficacious, both in the preclinical models, but also, of course, in the phase 3 studies, which is really what matters. And it's a really efficacious in preventing both disease and severe disease. And the safety has been acceptable. And basically, really, we see that there is a little bit more reactogenicity following the second dose. But most of the adverse events are between mild to moderate and really last for a short period of time. So we think that this vaccine, and I think we are seeing it both with the Pfizer and Moderna and the other vaccines that are really being used, has really the potential to address this pandemic. So I'll stop by really mentioning that there was a lot of groups that have helped and collaborated. This is something that would have not been possible without this extensive number of collaborations. Both with the NIH and VRC, the group of Barney Graham, Rob Cedar for all the preclinical studies, but also the funding from SAPI, BARDA, and support from NIH, and also the work with all the clinical sites and all the people that really joined and participated to the clinical studies and allowed us to demonstrate the efficacy of the vaccine. So I'll stop there, and I'm happy to take questions if there are any.
Thank you, Dr. Carvey. I'll start with a question, and I'll open the floor for the questions if someone has it. My question is that this is one of those first vaccines that, because of the pandemic, had to be rapidly make it to humans, and you were running, or your team was running preclinical studies, phase 1, phase 2, phase 3, and regulatory stuff. What were the challenges you all faced getting it to human?
I mean, I think
question, but
yeah, I mean, I think it's of course, the challenges changed during the process, right? I mean, clearly at the beginning, we did not think that this was was more of an exercise on how we could quickly develop a vaccine. As it became more clear that really the severity of the disease became even more urgent to develop the vaccine quickly. I think there were challenges, in both in terms of timelines. For starting the different clinical studies, you needed the preclinical data and generation of those data. We're on the critical path, and you have to have access to non-human primates. You have to demonstrate the safety and tolerability. So I think one of the challenges was that there are many moving parts, and you have to make decisions and take decisions sometimes at risk. Not a risk for people, because the safety of people that joined the clinical trial was really the top priority, but really trying to make sure that you don't make mistakes, right? And that you trigger the next step in your development at the right time, but as soon as possible. And I think, there were a lot of challenges and a lot of people involved. I mean, it's of course clinical research. But I think the most, more than the challenge, I would like to emphasize the collaboration, because everyone really tried, both within Moderna, but even outside, and again, the regulatory agencies, to help and to streamline the process and to really make the best decision possible, keeping in mind safety of people. So that's what I...
Thank you. Thank you for that. If there's no more question, we would make a move to the next talk, which is from Dr. Sanjay Singh. Dr. Sanjay Singh has received his doctoral degree in biochemistry from Central Drug Research Institute in Lucknow, India. Later, he joined the National Institute of Allergy and Infectious Diseases at NIH and headed the antigen research section for their malaria vaccine unit. In 2006, Dr. Singh founded the Genova Biopharmaceutical Limited in Pune, India. Under his leadership, Genova Pharmaceutical Biotechnology is dedicated to the development and commercialization of safe, efficacious, and affordable biotherapeutics. He led the successful commercialization of 7 life-saving biotherapeutics in cardiovascular, neurology, nephrology, and oncology market. Additionally, in partnership with various national and global organizations, including NIH, Genova has committed their R&D effort in vaccine development against malaria, cervical cancer, leishmaniasis, and tuberculosis. Today, Dr. Singh is going to talk about their efforts in developing self-amplifying mRNA-based vaccine against COVID-19. Dr. Singh, it's you now.
Thank you. Is I am audible properly?
Yes.
Thank you. So first, I would like to thank organizers for organizing such important meetings where they have brought one of the best people on this platform, and where the leaders from UPenn and Moderna, they are here. So they have made my job much easier to introduce it. But again, going back on the subject, this such a humongous problem of COVID-19 certainly gave, in difficult periods, certain opportunities like mRNA vaccine to be rethought. And the outcome has a successful vaccine from Moderna and a successful vaccine from Pfizer. So here in Genova, we started this program around 3 years before, but our primary aim was in personalized cancer vaccines, where we were trying to optimize on the mRNA side and also on the delivery side with our partners, which is HDT based out of Seattle. And as Pardy also said, and Dr. Karfi also said, the biggest problem is the delivery system. So here some innovative ideas came into our collaboration. So on the first side, we have gone for self-amplifying mRNA because we wanted to give a very low dose, either of the vaccine or therapeutics of the mRNA. And then we have also done some innovation on the delivery side, which is basically the nanolipid carrier. So here I'm going to focus how we are doing and where we are. So this is a cartoon for our program. And here the spike protein which we have taken, we have put into a self-machinery, which is basically self-amplifying mRNA. So what self-amplifying mRNA gives, that you have to inject a very low dose and it amplifies itself in the cytoplasm. I said that mRNA is very safe, it is not going to nucleus, so it is no chance of integration into the DNA. So it amplifies and make multiple copies. So you have to deliver a very low dose. But for delivery, this nanoparticle innovation basically came from our partners, HDT, and they have put a positive negative charge on the surface. So if you see this, when we combine these both the things, the mRNA is attached to the surface of the molecule. So this was what Genova we are doing. We are trying to add some innovation into biomanufacturing. And this gives us a freedom not to entrap the mRNA. And when the entrapment is not there, then we have a lot of advantage in terms of manufacturing capabilities. So when we were producing it, as Dr. Karfi said, our best known person in this area, Barney Graham, has said that how the hell you people have pulled out a 12 kb mRNA construct, because in non-self-replicating, your size is around 3 to 4 kilobases. And in our case, when we run this code, it is around 12 kilobase. So that was one of the challenges in front of us to make this whole system. But somehow we have succeeded and we are moving across. And how the results are that we are going to share with you. So basically the first was, as previously presented very well, that proof of concept, that how the design of the vaccine came, what kind of immunogenicity is coming in mice and what kind of immunogenicity is coming in non-human primates. And then the biggest challenge for here in India for us is how we scale it up, because scaling is a big thing and we were the early starter as compared to our peers like Moderna or Pfizer. They have a couple of years of experience, tens of years of experience into this area. So the scalability was this. And the third thing came out that we have gone through one preclinical toxicology, because this is the first time we are bringing mRNA in the country. So regulatory authorities asked not to do one, but they said before going into clinical trial, you have to do 2 preclinical toxicology. This was a difficult decision, but you have to go through regulators because they are doing in interest of people and interest of overall science. So we have completed that. And then we created the CGMP batches at a bigger scale, and for which we are audited, because again, this was the first manufacturing facility in India where the audit came out to ensure that is our clinical manufacturing practices are all right. So CIDISCO did the clinical, the manufacturing audit and gave us permission to go for clinical trial. Now I will go one by one into this. So this was the first proof of concept, was that we made this construct and injected 2 different regimes, which one is 50 microgram, another was 250 microgram. 250 microgram was a single dose, while 50 microgram was boosted on day 28. And if you see the titer finally skyrocketed, but they reached at the same point. But when we came back to check it, that how it is doing, in the pseudovirus, we got a very good neutralization assay. But if you see from the true virus neutralization assay on day 28 and day 42, we got a very good neutralization titer at that time. And this work was done at University of Washington back in Seattle. So this is the very first proof of principle it gave to us that 50 microgram and 250 microgram is doing very well. But in the meantime, we also wanted to ensure that how overall these non-human primates are doing in terms of the different parameters, which we have mentioned, many of the parameters here. And we found that clinical chemistry is fine, body weight is fine, there is nothing unusual has happened. So we were very happy with this data. And we published in this paper. Now here you want to say that, I want to mention here, this is 9,000 kilo base, 9,000 base, and this was the maximum ladder even available in the market. So we work around, and my team led by Ajay, we work around, and first we created a ladder which is 12 kilo base. And finally you can see that these are different concentrations we have loaded, and that looks so good and so beautiful. Then the complexing, if we talk about the complexing we did, so this is simple mRNA, where we complex, it goes on the top, and then we can extract it out also, mRNA gets released, and we develop methods to quantitate it. But in the next one you go that, what is the advantage of this technology? That here mRNA is very highly protected. So if you have this mRNA lane, you can see the lane this, but if we treat with RNAs, it disappears. But when we complex it and treat it, this is the lane, it doesn't affect at all. And even when we extract it out, you can see a very little loss in this. So this was one of the advantage we got on the size, that we can make a much bigger mRNA, which can be delivered at a very low dose. And this is the method, you can see how clean peak, like protein, we developed the method by like this, like protein peak, you can see and quantitate. But when we complex it, you can see a beautiful peaks comes out. And this, one of the advantage we created by doing this, we can do terminal filtration, terminal filter sterilization we can do, and which is unusual in the case of nanolipid particles. So this is one of the advantage we got. And when we put these things into the CGMP batches into our mouse, and we tried 0.1, 1, and 10 microgram, and you can see that when we have hit the 1 microgram or 10 microgram, even in 0.1 microgram, we hit the rooftop, means there is no scope going further on this. So this was one of the beautiful things came that we can go at a very low dose, and these are the antibody titer against spike protein. And if we see against RBD, the similar kind of profiles come, we have touched the rooftop, and this is very unique we have got. Because what I know at present, with the approval of a couple of vaccines now, there are clinical correlates of protection, and at present, there are two important clinical safety is very, very important, but the clinical correlates of protection in mRNA technology is very high titer as compared to other technologies. And then the neutralization assay, and this titer is staying. So if you see in case of ours, even 43 days, when we are looking at 57 days, even it is moving on the other upper side. So this was a very good news for us, because this is additional technology when we can filter sterilize 0.22. So it is not giving that kind of problem which can happen in nanomedicines, that there is particle formation in the finished formulation later on. We use the surrogate assay, which is the CPAS assay developed between Duke and Nanyang University. And here we got in our 43 days and 57 days, almost 100% at 1:10 dilution as their protocol. And when we tested in the print assay, this is unique, we are getting around 800-fold at 50, and the print 50 value at 800-fold dilution, which is unheard of, and we were very happy with this data. So basically, we produced the mRNA, and we have produced at a scale. We have characterized it very well. We tested into mouse, we have tested into monkeys, and it is passing all the criteria. And one of the advantages, today we are not able to produce the data to you, because the slow release of slow manufacturing of mRNA inside the cell which is, you can say, self-amplifying. We are getting some very positive data on Th1 cells, also Th1 cytosine response, and also looks like we will have a lot of advantage in inducing the germ center, which is the critical for long-term memory cells. So we have also there is correlation between antibody titer and potency. There are two challenges why we are taking time and slowly we are moving it. Because in environment like India, even if mRNA vaccine is very successful, it will be very difficult to ship it in India and go in middle-income countries if we have to ship at -70 degrees centigrade. We commend the effort made by Moderna and Pfizer, unbelievable success. They have opened the field in the direction of mRNA. But to have applicability in countries like India and other countries, many of the countries, we have to bring somewhere 2 to 8 degrees centigrade. This was one of the deployability was challenge for us, and why we have chosen a different technology. And the second is the limited portion for us, that very limited large-scale manufacturing of enzymes are available. So we have addressed both the questions from the day one. So these are the 5 enzymes which are basically, you start from plasmid, you need restriction enzyme, you need T7 of RNA polymerase, pyrophosphatase, RNA inhibitor, DNase, and capping enzymes. And they were all imported from outside. This is one of the big challenges for doing this kind of research here. And what we did, we put our efforts in other direction, and today we have 4 of the 5 enzymes we can manufacture here at Genova with us and with our partners. So this was another challenge in front of us for large-scale manufacturing, because we feel the mRNA technology is going to go forward, and also equally important is that we have to develop infrastructure inside the country to do it. We have at present installed.
Time, Dr. Singh, may you please wrap it up?
In a minute. So,
yes.
So we have around at present installed capacity of around 50 million doses that we are putting ourselves in, which we can go into 1 billion doses a year. That is the next part that we are doing. So just to sum up this up, so we have used D614G, and we are also working on the mutant version because this is the backbone for it. We are going for trials from 5 and 10 and 25 microgram. And then we have unique absorption chemistry, which is protected, and we have already stability demonstrated for 60 days today. So this is the team which is here at Genova, but very thankful for our collaborators, HTT, because without their help, it has not gone. And the most important, I would like to say, very thankful to DBT. If they would have not come for the support initially, they were very encouraging. And they supported this program. It was very unique. And then Indo-SAPI also funded. And the last we got funding from Mission COVID Suraksha. Our partner has got funding from NIH for doing their clinical trial in the US. Thank you very much.
Thank you. Thank you. Thank you for a very nice talk on the self-amplifying mRNA. Now we are going to switch the gear. And in the interest of time, we won't take any questions, and we'll move on to the next topic, which is the regulations and the regulatory agencies. The first talk will be given by Dr. Phil Krause. Dr. Phil Krause received his MD from Yale University School of Medicine. Currently, he's serving as a Deputy Director of FDA Office of Vaccine Research and Review. For the past year, he has also served as a chairperson for Research and Development Blueprint of the World Health Organization's COVID-19 Vaccine Working Group. In these capacities, he has helped to guide COVID vaccine development around the world by helping to set standards both for preclinical and clinical development of these vaccines. He co-led the regulatory team that recently authorized 2 mRNA vaccines and 1 adenovirus vector vaccine for emergency use in the United States. Besides his regulatory role at FDA, he's also a leading herpes virologist. His laboratory has made significant contribution to our current understanding of molecular mechanisms of latency and reactivation of the virus. Today, Dr. Phil Krause will offer his perspective on balancing speed and rigor of vaccine evaluation in context of global pandemic. And later he'll be joining by Dr. Somani for a panel discussion. Thank you.
Thank you very much, Sita. I really appreciate your invitation and as well as the work you did putting together this excellent group of talks, which really I think have been very informative for the audience. And I also learned a few things. So I would like to, before jumping into the talk, thank colleagues at FDA, WHO, and members of WHO expert advisory and working groups who selflessly contributed time, energy, and motivation for the ideas I'm presenting. And while I've stood on the shoulders of giants, if I should stumble, it's 100% my own fault. Oops. So COVID-19 vaccine development has been different than anything the world has ever contemplated doing. Many vaccines are being developed at the same time. And as these vaccines were being developed, we didn't know whether any of them would work, or whether some of them would work, or whether all of them would work. But as it became necessary to think about the criteria for success, the danger that a spurious result might occur where a vaccine that was actually not truly effective might, by random chance, turn out to appear to be effective was a very real concern. And in particular, this was a much bigger concern because many vaccines were being developed at the same time, which then increased the statistical probability of an outlier showing efficacy where it didn't actually have it. We also had the need for multiple vaccines. But along with that need for multiple vaccines, we didn't want weakly effective vaccines to interfere with the evaluation of better vaccines. We had a very specific safety concern for enhanced disease. And this was because based on the experience with respiratory syncytial virus, where an inactivated vaccine had actually led to the death of some children who received it and subsequently were infected with RSV, as well as animal studies with other coronavirus vaccines that suggested that this might be a possibility for coronaviruses, there was this concern that a vaccine could actually make the disease worse, either if the immunity was too low or if the immunity was of the wrong subtype, or subsequently as immunity wanes. And overall from this experience, the decision was made that vaccines should show a Th1 biased T-cell response, which would reduce the likelihood that this would happen. But of course, highly efficacious vaccines would be much less likely to have this happen also. A need for reliable data was obvious because we expected and have now seen very high deployment rates initially, so many people would be immunized before a mistake became apparent. From a manufacturing perspective, where there are global questions about availability of filling capacity in vials and syringes, if manufacturing resources were expended on the wrong vaccine, that could impact the ability to manufacture other vaccines. And of course, all of this was happening in the middle of a pandemic with a high level of scrutiny and desire for rapid results. So really, we had two major goals, and this is how the WHO thought about this initially in February a year ago. We needed confidence in vaccines in order to be sure we were giving the right vaccines. And this, of course, would also deal with, to some degree at least, with the issues of vaccine hesitancy. But we also needed speed because of the pandemic and the devastating effect it was having around the world. So we wanted the right vaccines promptly. And a key point there then was that the desire to rapidly deploy the first vaccine shouldn't keep us from finding the right vaccines. So what are the right vaccines? Well, they need to have sufficient efficacy. We hope it will last long enough. There should be efficacy not only against mild disease, but especially against severe disease, because this is a virus which is putting people into the hospital and killing people. It needed to be effective in relevant subpopulations, especially the elderly and ethnic and racial minorities who seem to be more likely to get serious disease from this virus. And of course, we needed a precision around the efficacy estimate that would support rational decision-making about vaccine use. It needed to be safe, with sufficient duration of follow-up and a large enough population to support deployment in billions of people, and of course, some evidence that it wouldn't cause enhanced disease. And then it needs to be deployable, which meant that it would be possible to consistently manufacture, and that along with that goes the fact that it needed to be sufficiently stable to distribute around the world, it needed to be possible to implement it as part of major, very large vaccination campaigns, and it needed to be possible to make enough of it that it could make a difference in a worldwide pandemic. And so supplies were very important too. And so these are the factors that went into the WHO's initial target product profile for COVID-19 vaccines. The WHO went further, and along with the FDA and some other regulators, defined efficacy criteria that would be needed for a vaccine. This included a 50% point estimate with a 30% lower bound. This would assure public health benefit, and in addition, likely a greater impact on severe disease. And we've noted that viral vaccines that are effective against all disease have also prevented transmission, assuring that clinical trials of adequate size to assess other critical outcomes would be done. And this was a point of great contention initially, where many of the developers, not surprisingly, wanted to be able to assess the vaccines with a 0% lower bound, on the theory that it would be easier to get a vaccine through. But of course, being sure that the vaccine really works is important. By reducing the risk of disinhibition, vaccinated people then might think they're protected, but it turns out that they're not, and then they end up spreading more disease. Reducing the risks associated with evaluating many vaccines, reducing the risk that the first vaccine would stymie development of later, better vaccines. The point was made that weakly effective vaccines would be much less likely to induce herd immunity. We really had one chance to get this right, at least initially. A weakly effective vaccine could do more harm than good, but also wouldn't have sufficient public health impact to justify rushing it. And so the lower bound of the 95% confidence interval really was a critical marker that provided substantial protection regarding the true efficacy of the vaccine that would be deployed. And I'd like to say that obtaining results quickly isn't a good thing if it shortchanged other critical evaluations: the safety database, efficacy in subpopulations, efficacy against severe disease, the potential for enhanced disease, and the duration of efficacy. And so the WHO was very clear that emergency authorization of vaccines should be based on at least 2 to 3 months of median follow-up. That vaccines should ultimately be followed to the primary endpoints of the trial, even if they had early efficacy and interim results, pointing out that early deployment of vaccines might interfere with getting all the needed data. This slide shows the risk that one takes if one has a lower bound that is too relaxed. And to the right of the curve here, you see that if you have a lower bound that is too relaxed, for instance, 0%, look at the triangles, you actually have a lower chance of detecting a truly effective vaccine than if you use a more rigorous lower bound, because the more rigorous lower bound means that you'll also have more people in the study. But of more concern even is that if you have a less rigorous lower bound, a vaccine with true efficacy around 20 to 30% might actually have a chance of being declared successful. And this is based on a need for a point estimate of 50%, but this is the risk that comes of not having large and rigorous enough studies. And in fact, we've seen this where not everybody has followed the WHO recommendations, and some of these studies have had fewer people in them and have not been designed as rigorously as what the WHO recommended. And this has led to some questions about the true efficacy of these vaccines. The point that the WHO also made was that in a trial with expected enrollment, you would get the results very quickly anyway. If the true efficacy was high, the first interim analysis would demonstrate efficacy with very high probability, even within a few months of starting enrollment. And we notice that the ability to detect efficacy increases with additional time. But this also shows, if you look at the difference between these 3.7 months and these 5 months, once the trial is enrolled, the data start coming in quickly. And what we found in reality was that these trials enrolled quickly, but then the rates of COVID-19 were even greater than expected. And so the actual results came in even faster than the WHO predicted in this slide. So what happened then? Well, let's look at the mRNA vaccines, which ultimately showed very high efficacy against COVID-19. To the left here, we have the Moderna vaccine. To the right, we have the Pfizer vaccine. These are the total number of cases in the primary analyses. And these are the curves over time, which you see for both of these vaccines, starting to separate around 12 days, with actually a fairly sharp separation, which suggests that vaccine efficacy is actually beginning quite early. And what's interesting is that this vaccine efficacy is detectable this early, but this is before there have been significant neutralizing responses by these vaccines, suggesting that it isn't necessarily just neutralizing antibodies that are protecting people. Now, the other thing is these are all two-dose vaccines, but you already see protection after the first dose. This doesn't mean that people don't need the second dose, because that likely contributes to prolongation of the immune response. We don't have enough people in here to tell, but it does tell us that a vaccine that is giving rise to these very rapid responses can have a significant effect in a pandemic, as indeed we've started to see. Adverse events were really not so different from many other vaccines. Injection site reactions, fever, headache, muscle pain, joint pain, and fatigue. And there were some questions about immediate type hypersensitivity, some cases of anaphylaxis, which have mostly been resolved with more follow-up, where the rates of anaphylaxis are roughly in line with what we've seen with other vaccines. There were early questions about whether these vaccines might cause Bell's palsy, which has not been observed in post-authorization follow-up. Other considerations include the age groups for which these vaccines were authorized. Pfizer down to age 16, Moderna starting at 18. The Pfizer vaccine had somewhat more complicated storage requirements, but either of these vaccines can be kept at room temperature for 6 hours after the vial is opened. Neither of them has a preservative, so it's important not to keep it at room temperature for more than those 6 hours. Dose volumes are a little bit different. The dosing regimens are a little bit different also. If we look at the courses available in 2021, though, based on the statements by the manufacturers, there would be enough vaccine made by Pfizer this year to vaccinate about 1 billion people, by Moderna to vaccinate 250 to 300 million. So this tells us that although these vaccines look like they're successful, this is not going to be enough people to vaccinate everybody around the world. So more vaccines are still needed, and it's important to keep developing additional vaccines. And we'll point out even vaccines with lower efficacy may have an important role to play if they have other advantages, for instance, in deployability, cost, number of doses, safety, duration of efficacy, etc. And indeed, the Janssen vaccine, which the FDA authorized over the weekend, has some of these advantages. And of course, the most efficient way to keep evaluating vaccines will be placebo-controlled trials. One can do non-inferiority trials, but they're much less efficient than placebo-controlled trials. They require roughly 3 times as many people or follow-up. So what does responsibility deployment of a COVID-19 responsible deployment look like? Well, vaccine needs to be rapidly delivered to those who need it. You need to keep collecting placebo-controlled data, especially to support safety and longer-term efficacy. When placebo-controlled studies can't be done, we'll need international collaboration on observational studies using many different systems to rapidly address safety signals, which we hope will enhance vaccine confidence and reduce the risk of spurious signals. An additional study will be very important to address vaccine effectiveness against emerging variants. So worldwide collaboration has helped to bring us where we are, and it's the logical path forward. A lot has been accomplished, and yet there's still also much to do. And the high observed efficacy of mRNA vaccines provides us with hope that we can soon bring life back to normal, either with the help of these vaccines or others that induce similar immune responses. So thank you very much, Sita.
Thank you. So now we are going to bring Dr. Somani, and Dr. Madhvi Rao from BIRAC is going to introduce Dr. Somani.
Thank you, Dr. Avasti, and it's an honor to introduce Dr. V.G. Somani. He's the current Drug Controller General of India since July 2019, and previously he was Joint Drug Controller of DCGI and was heading the divisions of regulatory, medical devices, in vitro diagnostics, stem cells and blood products, new drugs. He has also worked as Joint Drug Controller in the Division of Biologicals, Clinical Trial, New Drugs, Cosmetics. So over to you, Dr. Avasti, for moderating the session. Thank you.
Yes. Phil, are you still there?
Yes, I am.
Okay, good. So we'll start with Dr. Somani and back and forth with Dr. Phil. So this is the first time there are new vaccine modality has been introduced and it has to be introduced in a very short time. And so I was wondering the challenges that you faced in India, and then I'll move it over to Dr. Phil for that.
Yes, the challenges were definitely to look at who are the people who are coming forward, whether it is the universities or whether it is the manufacturers who are coming forward, which platforms they are coming forward with, and then the animal models and the assay methodologies, and what is their experience with those platforms. So this was a key challenge for India, because there were no people who work on whether this mRNA platform or DNA platform or even adenovirus kind of platform. So once these people have procured some technologies from outside, then the rest of the moments with regards to the animal studies, initial animal studies in mice, followed by the hamsters and non-human primates, they went on. And these were a very challenging time of the initial period. And thereafter, after going the things to the phase 1, then how to make it a adaptive trial design so that we can immediately come out with the results. And regulators had to do a lot of hand holding and inspections of the at the preclinical and clinical stages, preclinical or CMC stages during the development itself. That was the challenges in the initial stages.
Would you add something, Phil, regarding US?
Sure. Of course, one of the things to think about in a global pandemic is the emergency nature of the situation. And so yes, the vaccines have been made available in the US, and most of the drugs have been made available under emergency use authorization, which is a different standard from that which we use for licensure. And so there, it's the agency has to make the determination that the product may be effective, but however that the benefits exceed the risk. But for vaccines, we thought it was very important to look at this benefit-risk ratio in the context of a vaccine that would very rapidly be rolled out to many, many people. So your question is exactly what we were thinking about. And so that's one of the reasons why we insisted on a fairly high standard of clinical trial for looking at these vaccines, which included fairly large trials and significant follow-up. And I think it's because the big challenge is not only how do you look at the vaccine, how do you make the scientific determinations, but how do you build the public confidence in the vaccine that will cause people to take it. And so I think it's by, on the one hand, being able to employ a regulatory standard which is not quite at the same level of licensure, but nonetheless adapting that to the situation where we nonetheless required fairly rigorous trials and rigorous follow-up on people that are building the public confidence, that are causing people to get vaccinated. While not everybody who's eligible for vaccine has accepted it, the US has now vaccinated over 40 million people, and there continues to be huge demand for vaccines. So I have no doubt that over time, as people see what the vaccines are doing and how it's changing people's lives, the demand for vaccine will increase. But a lot of that is built on a solid regulatory foundation where people have confidence that this is being looked at in a way that is not perfunctory and is not cutting corners just because it's a global pandemic.
And thank you for that answer. And I would like to bring Sanjay Singh into the mix if he's around. Are you there, Sanjay? I guess he's not, but my question was that now with the approval of, oh, he's here. So the question that I was bringing to the panel, Sanjay, is that now with the approval of mRNA vaccines, what do you say as the future of other vaccine, other mRNA modality like self-amplifying mRNA? Not that you have to give a final concrete answer, but it's just what do you think of like self-amplifying RNA becoming as a new way of creating vaccines. To you, Dr. Somani.
Yeah. See, basically, presently what we, after the mRNA vaccines have established its efficacy, there are only two issues which we are now interested in. Its deployability in the situations like LMICs and countries like India, where the shipment conditions remain a very important factor. And if, as has been stated by the Sanjay Singh, if it succeeds, because they have given some stability data, because we have already seen it up to the 3 months of their vaccines at 2 to 8 degrees centigrade, and with that they are going. So this will be the one advantage. And with the self-replicating vaccine, mRNA vaccines, if the dose is reduced, then probably it would be possible to produce more and benefit more people with the doses. That is what we are expecting. So let us hope for the best. They are under the trial, phase 1 trial.
Thank you. Dr. Phil?
Sure. I just want to point out scientifically from what we've heard, although both of these vaccine types contain mRNA, they're actually quite different in their mechanism of action. In the case of the standard mRNA vaccine is developed in Drew Weissman's lab and as exemplified by the Pfizer and Moderna vaccine, these vaccines are using the methylpseudouridine backbone in order to reduce intracellular, especially type 1 interferon responses, which then could interfere with vaccine-induced translation and immune response. The self-amplifying RNA uses a different strategy. It's a little bit more like a virus itself because it makes more copies of its RNA in the cell. Those copies of the RNA don't have the methylpseudouridine, but it has the advantage of being able to make a lot more mRNA once it enters the cell as compared with the standard mRNA vaccine. So while both of these have mRNA in their title, these are different strategies. But I think that, as I pointed out, what we need still is a lot more vaccines. And the idea of having a vaccine where the overall dose needs to be lower and which has some advantages in deployment certainly is very appealing. But of course, because the mechanisms are different, it will still be necessary to do the clinical trials to prove that each of these strategies works independently of the others. And indeed, each of these vaccines, even the Pfizer and the Moderna vaccine, although they're similar in many ways, they also have some differences. They have different lipid nanoparticles, which could influence other aspects of the response. And so it's not possible to predict how one of these vaccines will work based on how the others fit.
That's true.
I think it's a promising approach, and I'm glad that Genova is pursuing.
Thank you. And I think our time is up, so I'm going to invite Dr. Madhavi Rao to close the session and thank everyone. Madhavi?
Thank you, Dr. Avasthi, and thank you all the panelists and today's speakers for this excellent session on mRNA vaccine. And I hope the audience and whomsoever is visiting this session would benefit a lot from this session. Thank you everyone. Thank you once again on behalf of DBT and BIREC. Thank you.
Thank you all. Thank you so much for joining this session.
Thanks, Madhavi, Sita, and the audience. Bye-bye.
Bye-bye.
Thank you, Philippe. Thank you, Sanjay.